This is a pretty quick lecture, I think. Mainly it's just to get you geared up to using some of the tools that are sort of follow-on tools once you have your gene lists of various kinds. And it's one of those, it's a problem that is not incredibly well solved yet, as any of you who have ever ended up with a complicated long gene list understand. Trying to make any kind of biological sense out of it is really the hard part. And very subjective, and the tools can help you point you to certain directions and so forth, but they're not going to solve the problem for you. So in the end, there's nothing for it. You've got to go to the literature and make up your own story based on some of these clues. But there are some nice tools that are available to help you see certain things that are good for displays, good for figuring out relationships between genes, and so forth. So I'll just talk really briefly about those, okay? So most of the methods you're talking about, ChIP-seq, RNA-seq, the intersection between ChIP-seq and RNA-seq, SNP lists, disease, variation sets, and so forth. They're going to give you a list of hundreds or thousands of genes. And so the question is, what do you do with that? And it's a huge problem that plagues us all. And so there is several ways to go at them. Aaron already told you a little bit about pathway analysis and GO categories. There are various kinds of tools for doing GO analysis. You've already had some experience with Gorilla and a few of these other tools. My favorite is one called David. It's just a really wonderful, very quick online tool that was created in order to analyze microarray data sets originally, but you can use it for just about anything where you've got a gene list or an identifier. And there is an exercise for using David if anybody wants to, but it's incredibly simple. And basically what it does is it puts things into GO categories. And if your experiment worked well, what you're going to see is that you got enrichment, really strong enrichment for certain GO categories. That's already a sign that everything worked well. If you get very high enrichment scores for GO categories. If you don't, it probably means that your experiment either didn't work well or you're working in an area of biology that just hasn't been annotated yet. That's the other possibility, that you're in an area of biology that just hasn't received a lot of attention. Because one of the things that's true about these categories that Aaron was talking about, and I think he's already said this, is that if the category doesn't have a lot of annotations in it, you're going to have a very small data set to compare against. You can never have good p-values, right? Not very reliable ones anyway. So as Aaron showed you, and as the Gorilla thing is structured, you can see that Go is a tree. At the top of the tree, the categories are very broad, and there's lots of genes. You can get really good scores in those. And as you go down the tree, you get into more and more specific categories. Those have fewer genes. Some of them have very few genes. There are some categories that have 5 or 6 genes in them. And so therefore, if you get one, you get a good score. And that doesn't necessarily mean that, that you're really in that kind of biology. So you have to just kind of be aware of all those things. But so Go and pathway analysis is really nice. David is kind of interesting because what it does is it collects Go, it collects some of the pathways. It's got KEGG pathways, it's got reactome, it's got all sorts of different things in it. It's also got protein domain databases, and if you hit any of those categories, it'll give you an enrichment. It'll tell you what's going on. So you can kind of put things together. Problem is it gives you these long lists of things, but David has a functionality, which is a clustering tool, which takes these categories and looks for similarities based on their gene contents. So as you know, one gene might have been annotated into blood development in one case and bone development in the other case because they were used in different kinds of studies. So you might get blood development and bone development and wonder what the heck is going on. Well, it may be the same genes. And if those gene sets have a lot of overlap, they can be clustered together so that when you get a category, you will see that both bone and blood development are in your category, in your clustered category. It may be also clustered together with the higher level annotations, like membrane bound things or secreted, and then the lower level things like secreted hormones. And then when you see all those things put together, suddenly your brain can click a little bit of a story together in terms of, okay, it's secreted and it's membrane, but it has to do with secretion of hormones and things that are influencing both blood and bone development. So what kind of things are those? David will also give you back the gene list so you know which genes have been annotated into all those categories. But this clustering tool is really nice, and that's part of what the exercise is for tomorrow, is to play around with using this clustering tool, which collapses some of these many, many categories into related entities that you can read into a story. And so that's part of what you'll get to play with. So that's what I just said. It gives you a really nice clustered score, which is based on the cumulative p-values of all the things that made it into that overlap set. And so it gives you a score, which is basically the negative log of the geometric mean of all of those p-values. And so that's sometimes kind of confusing to people because the score is a positive number. Like, a score of 10 is really, really a good score for enrichment of a cluster. But relating that back to specific scores like p-values is difficult because it's actually a cumulative score. So it's basically looking for all right, I have a set of genes that are enriched in all these different categories, but I can collapse them together into a group based on the similarity of the genes that are in those categories and give you a little bit more of a holistic sense of your data. So it's a tool that I find really interesting and really helpful, and there's a bunch of different kind of geo tools. I think the clustering idea is a particularly helpful approach. So, but what next? Which genes are most informative and representative in your cluster? So, especially for data sets with multiple different kinds of conditions and lots of different data points, you can use clustering algorithms that give you groups of genes that are moving together in your data set. So this is gene correlation, expression correlation patterns. It's the basis of WGCNA and all of the other things that Aaron was talking about. There are lots of different ways that you can use it besides WGCNA. But basically what you're looking for is across your data set, subclusters of genes that are moving together through the data set, either going up and then down or up and down and over. Now what you can use this for is if you've got a time course, or let's say different tissues, or lots of different individuals under different conditions. It doesn't work very well when you've got just a lot of different conditions, a lot of different people, for example, under the same conditions. But it works really, really well if you've got several different kinds of conditions, people that have been stressed versus people that haven't been stressed versus people, for example, lots of different conditions and lots of individuals, then you can use these clustering algorithms. But even if you've got a simpler data set, just mutant versus wild type, it can be useful to use some of these clustering algorithms. What you can't do is come up with a very good segregation of genes based on pattern if you've only got one condition and maybe three, like let's say you do a mutant versus wild type or a knockdown versus normal cells or something like that. But so the more data points you have in terms of different conditions and so forth, the better for these tools. So if you have genes that are moving together, and some of them a lot of them are unknown, but there's a gene in there that you know the function of that's got a pattern like this. It can indict the entire cluster of genes that are moving with it in a process called guilt by association. So if you have a set of genes that are moving like this, one of them is totally unknown, and some of them are known, you can indict them in the same functional space based on their correlation. So, and if you want to, let's say you have a list of 200 genes, and you've got them clustered into, say, 10 groups of 20 each, just theoretically, you don't need to look at every one of those 200 genes. You can pick examples from the 10 clusters to understand, to validate, to try to functionally explore. And so that is one way that you can reduce your gene set of 200 down to 20 that have something in common. But again, the more data points you have, the more different kinds of data points you have, the better discrimination you have in that ability. I wouldn't do it with just a couple of data points. So if you choose examples from the major clusters, you can help to analyze the whole thing. So cluster assignments are are the basis of what's called heat maps. You guys have all seen these. They make a really nice display for looking for genes that follow certain patterns up and down. And the input for a heat map or a correlation matrix looks like this, basically. So you're going to have genes, 1, 2, 3 to n. You're going to have conditions, so each condition and some value in the condition. So you can imagine this being CPM from an RNA-seq experiment. Or you can imagine it being something from a microarray, some sort of intensity value. You can imagine it being chip peaks. You can put all sorts of different things as long as you can associate an item, and it doesn't have to be a gene, it could be a location, with a score, you can do clustering. And so the score just has to be a numerical score that goes across a series of conditions, and then you've got your input for clustering. So it's a very simple thing to make. It's in the case of gene expression, they call this an expression correlation matrix. And or expression matrix, and you need to set this up, and then you can use that as input to go into these clustering algorithms. So it's very, very easy to do in general. But different kinds of statistical methods can measure pattern similarity based on this expression matrix. So things that track each other up and down through this path are going to be correlated. The simplest methods use a Pearson correlation statistics, but there are other variants like with WGCNA, which is a weighted correlation. It compares the vector of values for genes A and B over these conditions. And so this is the vector, and you're watching it go up and down over time, and you look for things that track it up and down. You can also look for negative correlations. So you can say when this one goes up, this one goes down. When this one goes down, this one goes up, and so on. And that can be also very useful when you're thinking about regulatory relationships. Some things are actually negatively correlated, and that can be extremely interesting. And most of these tools will allow you to have both kinds of correlations. Often you have to tell the program that you want to look at both negative and positive correlations, but it will do it for you pretty easily once you do that. And the larger the matrix, the more accurate you have in terms of discerning qualities. So there is a tool called Cluster, which probably a lot of you have used. If you haven't, you're going to get a chance to get your hands on it tomorrow. I think we're going to have you install it on your own computers, and so we'll take you through it, but that's good because you're going to have to install it on your own computers at home. This is a small piece of software. You can put it on any laptop. It is extremely useful for doing things like that. It's also extremely user-friendly. It has a lot more capability than we're going to show you tomorrow, and if you're really interested in it, it's got a manual about that thick. It's really, really a nice tool, and it's very well characterized. But basically what this has done is I've taken this expression correlation matrix of values and genes and given them intensity values. Actually, I think this looks like it's probably turned on its side. It is. So this would be the genes. These are the values over the genes. And basically, you can set the colors any way you want, but the usual pattern is red is high, green is low, and black is sort of not above the average. You can set this to do what's called centering, which is a nice way to do it, which says, give me this, for all these values that I've got here, show me the one, set everything at average, and then tell me what goes above and below. So that's a very common way to display heat maps. And that's how you get the red and the green there. You can log transform them so that you have a smoother map. There's all sorts of nice things you can do in the cluster tool. It's a super, super easy tool to use and very, very nice because it gives you these patterns. So now you can go back and you can say, these genes all have something in common. So do these, so do these. This little patch here has something in common. They have patterns that I can discern. But also cluster will give you a tree which shows you how they're related to each other as well. So that's something that's really nice to use. Now you can have groups, and you can actually extract the genes from those groups and use them as kind of exemplars of particular types of biology. So it was developed by Michael Eisen in very early days, and Latest version is Cluster 3.0. It's a very, very nice tool, very, very, very easy to install. The default is Pearson correlation, but you can actually set all sorts of different kinds of statistics. I wouldn't recommend you do it until you understand what the statistics do, because they all have different powers and problems, and you really need to know what you're doing. Although it's kind of fun to just play around with different statistics and see what the output looks like and just learn for yourself what they do. And often they'll give you a very similar network. It depends on the data and how complicated it is. But it's kind of fun. It's a GUI that you can just download onto your computer, and you can combine it with a tool called Java Tree View software, which is another very easy to download tool, and it'll make these displays for you. And it's very nice. It's an interactive tool where you can, click on a clade and it'll pull up the gene names and stuff like that for you. So that's a really commonly used tool. It's based entirely on expression correlation, as most of the tools are that we have. But like I said, you can enter anything where you can set up an item and a value across conditions. You can cluster. So you can cluster chip peaks if you want to, if you had a need to do such a thing. And that can give you groups of peaks. Get groups of, for example, if you've got changing conditions, groups of peaks that are changing over time and so forth. And it gives you a kind of a nice display if you've got the right kind of data set. I already talked about centering. That's basically setting the average and letting everything be green or red depending on whether it goes up or down for that particular gene over the data set. So it's just looking at the gene's pattern of up and down, basically, that way. So that means if you've got a gene that's expressed at really low levels and it's going up and down, and a gene that's expressed at really high levels and up and down, they're both set at their own averages. So you're just looking at the variation. And that is the most common way to use this tool. Correlation networks are also something that WGCNA is a much more sophisticated version than the one that you're going to have a chance to play with here. It also requires WGCNA, I would say, a little bit more study and understanding of the statistics before you use it, whereas this tool is really, really easy. Expression correlation networks is the thing that we're going to, again, download. You can visualize it in a tool called Cytoscape. Has anybody ever used Cytoscape? Nobody's used Cytoscape yet? It's really a cool tool, and it does a lot more than I can show you. I just want you to get a taste of using it. It's really neat. It's mostly for network drawings. You can load also, it has a little app store that you can go to, and you can download all sorts of plug-in applications. Again, in order to use them, you really need to read their manuals and understand how to use them. We're going to use a simple one called Expression Correlation Networks. It is basically the same correlation, Pearson correlation, as you use for cluster. But it displays things in a network view. And so basically what you can do is you can say, show me all the relationships between genes in a data set, and now show me their relationships to each other. You can color code whether the relationship is negative or positive. So it records those things for you. It does both sides of the correlation. And you can draw networks. It also gives you tools for looking at things like what they call centrality, network centrality. So basically, it's basically how many things, if you're going to go from this side of the network to that side of the network, how many nodes have to go through this guy? The answer is most of them. It can tell you what are the most central nodes in your network. That's what centrality means, betweenless centrality, for example. And there are other kinds of centralities that you can also monitor using Cytoscape. It'll give you tools for doing that analysis once you construct the network. So it's quite nice. The tool, expression correlation is one of the plugins you can put into Cytoscape. It calculates the network for you, and then Cytoscape can do all of the betweenness and so forth calculations. So it gives you information about the nodes that are most central, the ones that are most peripheral. It gives you actually a score for correlations. It's very, very nice, and it can tell you some things that can just be really good for hypothesis generation. This tool is really simple, so it's, and luckily they just have, for years it was not available on the latest Cytoscape, but it's just been upgraded, so it's really, really nice now that it fits in with the latest Cytoscape. So in a network, basically the distances between the vectors are is a similarity of the two genes, and so if they're closer together, they're more similar, and they're arranged all in sort of a way that fits the whole network together. So that's they end up being in a particular position because they're being pulled aside by this guy, for example. So it basically just tells you the distances and the relationships of nodes to each other. The more you can put in quite a lot of nodes into expression correlation, and you will get this, the classical hairball, which is usually you can't tell anything except structure from very far away, but Cytoscape also lets you zoom in on particular clusters, so you could look at this one separately from that one, for example. It lets you look for what's called the nearest neighbors, so you can click on this node and ask which ones it's most closely connected to, and sometimes it's not obvious from the display, and especially on a really complicated display, so it'll pull out all the most closely related nodes and let you make a separate network from them. So it's really, really a very nice tool. Like I said, expression correlation is one of the simplest tools, but it's also the easiest to understand, and it's just a really nice way to view big data sets. You will not get a good expression correlation network unless you have conditions, and so I just want to repeat that. If you just have 3 samples, you're not going to get an expression correlation network that means anything at all. You really need a lot of samples and a lot of different conditions to get a good network. Often you might want to pull out a subnetwork. Let's say you have 5,000 genes or 3,000 genes or 2,000 genes. You probably won't be able to view them all. So it's nice to be able to pull out a subset, and sometimes you can do that by just pulling out, let's say, the differentially expressed genes once you have the network, and there are ways to do that in Cytoscape as well. So it's good to use all the data you've got for the network, but then pull out a subset, and so you can do all of those kinds of things. You've already heard about this from Aaron, so this is just a really summary of what I've just said. This was from last year, and I didn't correct it. 2.8.1 is the old Cytoscape, but now it works on version 3, and that's what we're going to do tomorrow. It's basically for hypothesis development. So it's going to be some simple tools. The exercises you have is to create a heat map from expression data using cluster. Use the same expression data set to go into Cytoscape and make a network and analyze the network. And if you want to, you can analyze a gene expression data set from Geo and download an expression matrix file. So how many of you have ever heard of Geo? Yeah. It is where you have to deposit your data, and if you publish anything related to microarrays or sequencing these days, right? So everybody deposits their data there, and one of the things you do when you deposit data, most people do, when they deposit data to Geo, is upload a text file, which is the expression correlation matrix. So you can download that and analyze it if you want. If there's a data set you like, or you're interested in, you can download that data set, the expression correlation matrix. You can tailor it to suit. Often, for example, most of them will report even if there's zeros all across. You can take those out and remove things with low values. You can average over the data set and only pick out the things with the high expression values. You can do whatever you want to the expression matrix. It's just an Excel, a tab delimited file that can open up in Excel. And then you'll have a data set that you can analyze with these same tools. I don't think you're going to have time to do that because you all want to get onto your projects, but if you want to do it at home, you can do that. It's a really nice thing to do. And you can also select a subset of genes from a genome-wide expression matrix using Galaxy, so you can go in and just pull out a subset of genes that you want to display in a network. And I have instructions for how to do that as well. So that's just to introduce you into what you can play around with if you want to tomorrow. Basically, the exercise tomorrow is just your choice. If you want to learn a particular thing, you can do it. If you'd rather get to your projects, you can do it. You may want to use some of these tools for your projects, and they'll be there for you later. So it's really your choice tomorrow. We want you to have some time to work on your projects, and so that's the most important things. But several of you are doing things where you might want to use some of these tools at the end of your work, And the instructions will be there for you to do. Also, when you go home, the instructions will be there as well if you want to play with them there, okay? That's it.